unSILOed podcast is produced by University FM, elevating the stories of your institution.
Welcome to unSILOed. This is Greg LeBlanc, and I'm here today with John Tregoning, who is a professor of vaccine immunology at Imperial College in London, and also the author of this book, Infectious, Pathogens and How We Fight Them. Welcome, John.
Hi, thank you. Thank you for having me on.
So look, this book, it came out, I think, in 2021. I think you put the finishing touches on it in right at the end of 2020. And, I think this book deserved to be read by pretty much everybody, because what you did is you managed to take all of the things that people were obsessing on, right, which had to do with microbiology, right, immunology, epidemiology, which they were getting sort of a drip feed of this stuff right through the news media, but you managed to put it all into a very concise and, I should say, entertaining package. And, it's hard to make these topics entertaining, but somehow you managed to do that. And I think, one question I would have is, do you think that the pandemic has raised our awareness of these issues, I mean, among the general educated public, to a level, where it'll get continued attention? Or do you think this was just sort of a blip on the radar, and now people will go back to their daily business and sort of, forget about these topics? Because I think towards the end you were saying that, if we want to keep the fight against pathogens and infectious diseases and parasites and so forth, if we want to keep making progress, then we have to keep investing in it. Do you think that the investment and attention is kind of cyclical? We're fighting the last war, or do you think that, we're now at a place where people appreciate the need to invest resources in this?
I think if we start on the knowledge and the awareness, I think that is the conversation is greater. I think people are talking at a more informed level about vaccines, RNA, antibodies. The awareness of those kind of words is much greater, because the R value, the R naught, how diseases spread. I think because it was so concurrent and people were discussing it all the time, I think a general awareness of these ideas is better than it was 5 years ago. And the fact that, there can be a respiratory pathogen pandemic. If you'd asked people 5 years ago, they'd probably say, no, I don't think that's going to happen. It wouldn't have been high on their list of worries. And now there's much more awareness that these things exist and these things can happen. In terms of funding, I think possibly it is going to be cyclical. And I think people will feel that there may be a general sense that, that's happened, we're not going to see another one for our lifetimes. I think that's a mistake. I think we're very likely to have a flu pandemic in the next 5 to 10 years, because those happen on a much shorter cycle. So I think maybe the funding hasn't followed the interest, or the news has moved on, and people are interested in other things. And there are other, and that's probably right. There are more pressing things that people should be worried about or talking about in going on in the world at the moment. But so there's a bit of a balance.
Well, look, I mean, the book is also kind of a history, right? So it's a Whig history, right? Because it's all about progress, and but it's relatively recent. I mean, you do go back to like Leeuwenhoek, but fundamentally the history of this dramatic decline in mortality due to pathogens, it's really a modern history, right? I mean, Jon Snow, I guess, is probably the oldest real hero in the book in terms of moving the needle forward. But even though it's a relatively recent history, I think most people prior to the pandemic, at least in the modern Western world, were kind of oblivious to this fundamental transformation in terms of, how we had gotten disease and of this sort under control. I mean, the statistics are crazy. I think, the majority of people probably died of some infectious disease, you know, in the 19th century. And now, even during the pandemic, right? Even during the height of the pandemic, most deaths were due to, you know, non-pathogenic reasons. I mean, is that transformation something that you know, for some reason is just sort of forgotten? I mean, why are people aware of how badly the world wanted us dead, you know, prior to the 20th century?
Yeah, and it's not, I think it's more recent. If you look at the UK data, it's kind of the 1950s is the tipping point
Mm.
from communicable to non-communicable diseases. I, it's almost, I feel like it's a bit of like, if you don't see it, it doesn't exist. It's a sort of, the absence of these things killing people has made people assume they no longer exist rather than they're no longer a threat, which are very different things. And the the generational change, or even things like polio, that that from the 70s to now, it's a huge difference. And the number of people who kind of quite famous people from that era who had polio and grew up with the consequences of polio, and we don't see that now in the like you say, in the Western Hemisphere. So I don't know why the attention has slipped, or the kind of the lack of awareness. Some of it, and I've been thinking about non-communicable disease now. Some of it is maybe it's because it's a zero-sum game. You have to die of something. So infectious diseases was replaced by heart attacks, and then heart attacks has gone right down. So that's replaced by cancer, and that becomes the next big thing. So maybe it's always just about the thing that your nearest and dearest are dying of, and you don't worry about the other things. So I don't know why it isn't more, just, and it is amazing, I think that the kind of miracle cure, or the change for me, is in my lifetime, is going from Freddie Mercury dying of HIV to it being a treatable, not curable, but controllable disease. And that's going from me as a teenager to me as a 40-year-old. That's incredible progress. And but then maybe the success, it's sort of a victim of success, because it disappears from people's consciousness. It's curious.
But there is all this kind of combustible tinder out there, right? I mean, there's an arms race between us and the pathogens, and while there's progress, there's also kind of retreat. And so in the background, all of these nasty bugs are trying to figure out a way around all of our defenses. So I think that in the book, there's part of the book is really a taxonomy of the different pathogens, right? So you walk through bacteria and viruses and fungi and parasites. But then the other part of the book is really about how we kind of keep these things at bay, right? And vaccines actually represent, and the immune system itself, is sort of the almost like the last line of defense. There are a whole bunch of other things that we do, and in some sense, probably, clean water was probably the most important thing, right, that we have developed to kind of keep our health. So maybe we can walk through the history of microbiology a little bit, and talk about some of the heroes that helped us to understand more about these little tiny things.
Yeah, absolutely. So I mean, I guess we start with Leeuwenhoek, who you mentioned, who invented the first microscope, and I think that's a key moment, because I think you go from assuming all sorts of things cause disease, bad air, whatever it is, and I think some of the kind of the way people, if you couldn't see it, they were associating it with the right things. They're saying, don't, you get a malaria if you go to a swamp, or you get this disease if you go to a dirty river. So the sort of associations were there, and they were filling in the gaps as best they could with the knowledge at the time. And that's all science ever is. It's an imperfect answer. There's always doubt. There's always questions. And we're just using the tools we have at the time to interpret the world around us. But Leeuwenhoek finding that these, microscopic organisms existed open up the world to saying, well, maybe they're doing something. What are these things that live around us doing? And so that begins the kind of understanding of bacterial pathogens. And so you have two very important people in that kind of in the next step. And one is Louis Pasteur, a Frenchman, and the other is Robert Koch, a German. And they were contemporaries. They sort of overlapped during the Franco-Prussian War, so they were actually quite. There's a sort of history of conflict, both nationally and personally, between the two of them. Pasteur was the first person really to show that disease doesn't just happen. So it was assumed that you'd sort of spontaneously create an infection in your lungs, and then that's what makes you sick. And he said, no, there's a living organism, and if you keep that organism out, you don't get infected. And he showed this with basically a flask of soup that he sealed and sterilized, and if as long as you kept it free from the air, it didn't get infected, didn't go off. But if you let the air in and the bacteria in the air in, it got infected. So he was saying, look, this is the sort of germ theory of disease. The germs exist and they can cause disease. At the same time, or sort of fairly similar time, Robert Koch was coming up with his postulates, his ideas that said to kind of rules about infection and pathogens, that if you can isolate a bacteria or a pathogen from a person, if you can then grow it and then give it to somebody else and they get the same disease, that links it all together. So those two really kind of founded the bacterial field.
Now, but I mean, it seems like you don't need to have an understanding of the proximate agent to understand person-to-person transmission, right? So they had, quarantines and so forth. It seems like the more difficult thing for them to understand were these kind of vector-borne diseases, right? Because the channels by which they were transmitted, were maybe a little bit more difficult to understand, right?
Yeah, no, that's an interesting point. So Walter Reed, who was based down in the Panama Canal, was associated with understanding how yellow fever spread, and that was mosquito-borne. And actually, the way he did that was to take mosquitoes that had bitten one person and then do a human infection study, so deliberately expose another person to the same mosquitoes to show the vector. So yeah, no, it's interesting, like the conceptual idea that you can get a disease from another thing. I suppose it had been around from something like rabies. You see rabies bite, it'd be a bite by an animal. It's just bring it down to a much smaller scale. And a lot of the virus work, was preceding the idea that viruses even existed. So bacteria are sufficiently big that you can see them with a light microscope, the sort of things that people might use at home with two lenses. But viruses are incredibly small, so small that you need either an electron microscope. But the people working on them inferred that they existed by using smaller and smaller filters, stuff that would filter out all the bacteria and yet retain the ability to infect. So this kind of absence of everything else. It's like, well, there's something in there. We know what it is doing, but we don't know what it is. And there was at least a 40-year gap between the first idea of a virus and somebody actually seeing one using one of these electron microscopes.
But it is interesting that people have to actually see it to really believe it. I mean, when you look at what Semmelweis did, right? I mean, it was almost like a clinical trial. I mean, he demonstrated the efficacy of hand washing, but it wasn't enough really to get people to adopt the principle. It was like you needed to see the actual, agent at work, right?
Yeah, because he preceded Pasteur and the germ theory of disease. And I wonder if some of it was communication, like ideas. It was much harder to communicate ideas. They'd be in a native language. We are so blessed now that most sciences communicate, certainly as an English speaker, blessed, that everything's done in English. So I gave a seminar in the Netherlands a week ago in English, and the whole thing was in English. And so the ideas transmit. The speed of ideas transmission now is so much faster. And maybe if Semmelweis had been doing his kind of clinical trial in a period where there were enough people to pick up on his ideas, it might have spread further. So I think there was a bit of that. There was a bit of resistance from the community who he was essentially saying that your practice is bad and nobody wants to hear that. And then you're right, there wasn't this kind of additional layer of like, and this is why the thing I'm doing works.
Mm-hmm. And I think once people discovered antibiotics, we kind of went crazy with them, right? I mean, one of the things that I find fascinating about the last couple of decades is this kind of movement back towards an appreciation of the microbiome, right? So that and do you think that, I mean, the people who study these, are they divided into the folks who are disease fighters and the other folks who are more interested in bacteria generally as a positive or a negative? I mean, because I mean, I know there's science and then there's engineering, right? There's science and then there's medicine. There's the folks that are interested in just understanding the phenomenon, and then there's the folks that are interested in developing tools to kind of improve the human condition. Is there a similar dichotomy in the world of microbiology?
Yes, I think so. I think you could tend to split people into camps based on whether they're basic scientists or translational scientists. And basic scientists are the one, and it's very crude, but basic scientists are thinking about just fundamental questions that may never go anywhere. They are interested in, why does this particular protein do this particular thing in this bacteria, and addressing that sort of knowledge for knowledge itself. Often that is the seed stock that then leads to the thing down the line. If you know that the bacteria makes its cell wall in a certain way, you may be able to then design a drug that fits into that enzyme. And then there are, like you say, translational people, and they're probably closer to the engineering, problem solving, thinking about how do we discover or how do we make new drugs? How do we treat these problems that we have? And so you get a spectrum of people working across the piece, and you get some people who do exclusively one or exclusively the other, or overlapping between the two. And you see that in immunology, you see that in microbiology, you see it in virology, and some of it is I guess it's the club that you put yourself in. So if you're a virologist, you tend to think about the world work-wise. You think about the world from the virus's perspective. I'm an immunologist, but I work on viruses, so I think about the host response before I think about what the virus is doing. So there's a bit about kind of the club you're in and the people with whom you share your research.
Yeah, and I was fascinated by the extent to which people who oftentimes kind of flamed out in academia, migrated over into the for-profit sector, right? So there were a lot of folks who were making advances at, say, pharmaceutical companies. And is that within your community? I mean, if you're working in a corporate lab, is that viewed differently as working in an academic lab?
It's the way the focus is driven. In the corporate pharmaceutical world, the resources are so much greater to solve a very specific problem. It's to say, how do we make a vaccine against X? And every question will be focused, every experiment will be focused on how to do that specific thing. Whereas an academic lab, it might be, what does this problem mean? How does RSV get into cells? I think it's more fluid between the two communities now, and I think that the respect from both angles is much better. It used to be, like you said, you've fallen, or some people sort of nearly say, "Oh, has gone to the dark side," as if the science is different, but the thought processes and the integrity and the robustness of the work done in the big pharma companies is every bit the same as in academia, and probably on some levels it is much more strict because it has to be recorded and it has to be to get everything exactly right so it can go through to a product. It just has a different endpoint to maybe the kind of pure blue skies basic science work. And people are moving between the two, and with the beginnings of these smaller like biotech companies, they're closer to academia. So I think there's a whole range. There's places in the research kind of ecosystem for everyone, no matter what their kind of personal approach to sciences.
Now, during the pandemic, we learned a lot about B cells and T cells. But I think one of the points you're making is that, by the time you get to the B cells and the T cells, those are kind of like the last lines of defense, right? If you get to that stage, then, you've got a bunch of different failures all along the way. So, when you're thinking about kind of disease prevention, presumably there's an ROI to all of these different types of barriers that you can put between you and the pathogens. Where is the best ROI? I mean, I think during the pandemic we were talking about behavioral modifications. We were talking about environmental modifications. We were talking about different kinds of immunological modifications like vaccines. I mean, how do we know in the world of public health, where we should be investing our resources?
So I think some of it, one of the models I really like when people were talking about it was the Swiss cheese model. So if you imagine a piece of Swiss cheese with lots of holes in it, each piece is an imperfect barrier. But if you start lining them up and you put enough layers of Swiss cheese, eventually there's no pathway through for the pathogen to get from the outside into the body. And the basic measure is that more public health, the bigger every pound spent on public health saves £2 later down the line. So if I had the big pot of money, I would be investing in people's basic underpinning health. So make sure trying to reduce the obesity, trying to reduce smoking, trying to reduce drinking, all of the things that make it worse when the pathogen gets to you. All of that investment is going to pay double because it pays you in not getting infected, but it pays you in kind of overall general health as well. So the earlier you can nip it in the bud, the better, I think. And so that goes for prevention. It probably goes for thinking about surveillance, investing money in making sure that we're catching the pathogens early, and then having high containment units. The original SARS-CoV-1, admittedly spread in a different way, but was contained because there were high containment health infectious diseases units in the countries where it broke out. So it didn't spread through the population. So there's a lot of public health investment before which if you get right, you avoid ever needing to think about antivirals and vaccines and the things further down the piece.
Right, and I think probably your prevention or intervention strategy is motivated by your causal model, right? So I think a lot of people are frustrated when we have these kind of unicausal models, right? So if someone say gets infected with COVID and dies, or I should be more precise, right, with the SARS-CoV-2 virus and dies, we say they were killed by this, but you could argue they were killed by, and there's probably a dozen but-for causes, right? But for infection, yes, but for underlying health conditions, yes, but for walking out into a public space, yes, and so forth. So attributing these kind of single causes to the ultimate outcome kind of obfuscates all of the different possible ways that you can, prevent that death, right?
And I was talking something about this recently. Each person, it comes down to how we have to do the science to think about it. We have to simplify it down to as basic a thing as possible to be able to put people into pots to make meaningful choices, to say, this person had X and therefore Y happened. But each person's exposure experience is completely different. The bacteria we carry on our skin is different. The meal we ate yesterday, the level of complexity of an individual person or even a cell is so great that our understanding of it isn't good enough to be able to cope with all of those things, to not simplify it down to a place where we can make sensible decisions. So I think you have to bring it down to single things in order to be able to do meaningful research and understanding and prevention.
Well, maybe we can talk about vaccines. I mean, vaccines are really remarkable because they, essentially utilize the inherent immune system to kind of nip off these problems in the bud. Um, is there? I mean, it seems like there were diminishing returns in the design and of vaccines, but we've kind of turned a corner now. Are we going to see sort of a renaissance of like new vaccines coming online post COVID?
I think so. I mean, I think, one of the most exciting pieces of news for me is that there's a vaccine for a virus called respiratory syncytial virus. So this is something that children and the elderly.
This is your area of expertise,
Yeah,
right?
yeah, exactly. So this is the thing I've been working on for 20 years, and for the last 20 years, or forever, but for my 20-year academic experience, children get it every winter. The whole intensive care unit will be filled with children sick with this particular virus, and then it goes away, and then it comes back the next one, and it just has been cyclical. It's a huge burden on the pediatric units. It maybe doesn't kill very many people in rich countries, but in low-income countries, it's a high burden of death because there aren't the kind of intensive care units to cope with that. So it's a childhood disease, but it's also in the very elderly. It's a significant cause of mortality. And there have been thoughts that there would never be a vaccine for it. It seemed like an unsolvable problem. And yet, here we are in 2023, and we have two new vaccines for the elderly. There's a vaccine for pregnant mothers to protect their babies, and there's an antibody, which is slightly different to the vaccines, which will also be usable in children. So it looks like the virus may not be so significant in the next 5-10 years, and that's a huge change. So, and that's it. It's kind of maybe was parallel to the work going on in COVID, but it shows that investment in vaccines is very effective. And the next kind of level of it is, do we start making vaccines for diseases that have long-term sequelae? So something like EBV, which of course we say glandular fever in the UK, but you say mono, I think. It's a kind of childhood disease, makes teenagers quite sick, but it actually goes away then, but it can cause cancer, and there's a very strong suggestion that it causes MS in people as well. Mm-hmm. So it hadn't been a vaccine priority because the primary disease wasn't that serious, but this link to MS, if you suddenly have a vaccine that stops MS, that's a huge protective and saving. So I think understanding that maybe no pathogen is good for you, and getting rid of all the pathogens through vaccination, maybe that has kind of re-energized the field.
Right. And so as an economist, I'm always interested in trade-offs. And so, you can have too much or too little of something. And so, I'm always fascinated by this idea that you can have good and bad, bacteria, and that you can use good bacteria to fight the bad bacteria. Are there any good viruses, or are all viruses bad? I mean, I guess there are, bacteriophages, which are I guess good viruses, right? Is that another area where we're going to see a huge amount of research?
Yeah. So they're good viruses because they don't affect us. I mean, they're not. So bacteriophage aren't killing us. So I guess we can see them as good viruses. Whether they, I'll come back to the first question about good and bad, but whether bacteriophages, which were historically used as like antibacterial treatments, and this was kind of through the 20s, it carried on being used, particularly in the USSR, because they kind of focused on that rather than antibiotics. So there may be ways to kind of make bacteriophage better at killing bacteria, but bacteria have had a very long time to evolve to fight bacteriophages. As long as there's been probably bacteriophage pre-exist bacteria. So this kind of humans and antibiotics is a blink of an eye in the context of bacteriophage and bacteria. The CRISPR-Cas system, the way that people are now engineering genes, is a bacterial antibacteriophage system. So bacteria have their own immune system. So yes, bacteriophage you could kind of see them as a beneficial virus. Is there a good human virus? I don't think so. I think there's viral stuff in our DNA which we evolved with so long ago that it's become part of the way our bodies work. Yes, that's probably important. There's integrated retroviruses that may be important. But I think if you could never catch a virus, would your life be any worse? I would go out on a limb and say no. I think you'd be fine. There's an idea that maybe you need some training of your immune system, but I don't know. They're parasitic things that cause damage.
Now, of course, people are worried about biodiversity, but there are certain species that we're not going to miss, right? So, I mean, of course, smallpox probably is something that no one's going to miss. But if are people seriously considering, say, eliminating mosquitoes, the ones that are the bad ones, right? The ones that, or if we were to would it be better to try and target eliminating the malaria itself?
So, yeah, I think that's a important question. So I think mosquitoes per se are an important part of ecosystems and will be eaten by the next animal up. But the Anopheles mosquito that carries malaria potentially could be replaced by any other number of mosquitoes. There's hundreds of strains of mosquitoes. So if there's only one that carries malaria, maybe the trade-off of eradicating that isn't so bad ecologically. But it's quite, yeah. I think that because they must be part of the food chain, that then becomes more complex.
I mean, with respect to antibiotics, I mean, do you think that we're just this is like a little blip on the historical horizon, where there was just so much, we compacted probably 100,000 years of evolution into like 100 years on the antibacterial side, that if we fast forward 500 years, I mean, do you think any of the antibiotics that we currently use are actually going to work in 500 years? Or we just have to we have to keep inventing new ones? Or is there some basic trade-offs in the bacteria that if we bombard them with 5, 6, 7 of them, there's no way that they could design a protection against all of them without degrading their ability to function in general?
The majority of antibiotics that we use, the vast majority, are all natural products. So they've evolved in another species to fight the bacteria. So the fact that bacteria are resistant to them isn't necessarily, or didn't emerge necessarily from human use. It existed because the bacteria were fighting the yeast that made the penicillin in the first place. What maybe has gone faster is that the spread of this resistance has become much wider. It might have been a local thing, and now it's become a kind of global thing. Will there be the same antibiotics? Potentially not. It depends how they're used and stewarded. So there is a cost to being antibiotic resistant. Comes with a certain cost to most bacteria. The just making the extra proteins and machinery to fight the antibiotics. If you're not being selected for that trait, the bug that doesn't have it can grow quicker because it's not making quite as many proteins. There's a sort of marginal gain in that. So the trade-off is that maybe if we nobody used penicillin again for 100 years, it might suddenly be useful in 100 years' time. So there's a sort of trade-off for the bacteria as much as there is for people.
Yeah, I remember I got a staph infection on my leg, and I went to a doctor in France, and he said, "Oh, I'm just going to give you this really old antibiotic, right, that no one never uses anymore," and the thing healed right up, and I thought, so I wonder, maybe what we'll do is we'll kind of cycle through these, right? So they'll become ineffective, and people won't use them for a while, and then 100 years from now they'll be usable again, and maybe we'll just be rotating through them.
Yeah, it's like fashion. Everything comes around.
Thick ties and thin ties, that sort of thing. Now, with respect to the immune system, I mean, is there any limit to when we think about the acquired immune system? I mean, with the inherent immune system, right, there's trade-offs. You can have too much inflammation or too little inflammation. But with respect to the acquired immune system, I mean, is there any limit to the number of pathogens that we can develop a memory for? I mean, is there a finite amount of memory? If you acquire immunity to one thing, does that mean that you have to kind of cut back on the production of others, or lose the capacity to produce others?
I think there's two bits. The probably the amount of antibody you can keep in your blood at any one time is set. So say it's 1,000 different antibodies per milliliter of blood. It's way more than that. But then if you get the next infection and that you make 100 antibodies to do that, you will lose the antibodies from the blood. Your antibody, if we measured your blood every day and we could possibly do a screen of what it's binding, it's going to change all the time. And that's the kind of effector bit. But that's a bit like it's saying the books that you've taken out of the library change at any one time when you're knowing what you need to fight off. The library itself, I think, is much, much broader, and probably you don't displace immunity. You probably don't need very many cells of memory cells to then expand up in the future. So the thing you saw in your childhood 40 years ago may have contracted right down to a tiny thing, but if you see it again, it can expand up very quickly. So I think at the effector end, the bits of the immune system doing something, there's a finite amount of that, but the memory part, people have explored it. It's possible it could be exhausted, or you could kind of fill it up. But I think even in just pathogens is such a tiny part of what our immune system's seeing. The vast majority of microorganisms our body sees are not pathogenic, and we'll have some kind of immune memory against them.
So the library metaphor is you have sort of a whole bunch of templates, right, that you can put into the factory, and on command, and then start cranking out the antibodies. But there's still lots of room on the shelf for more templates.
Yeah. There's gaps, and it can and things can expand to fill those gaps.
Mm-hmm. So another thing I think a lot of people are confused by is why some vaccines seem to be effective against the kind of all of the various mutations of a disease, and others seem to be much more limited. So, for instance, with smallpox and with polio, I mean, we're just eradicating these things. But with things like the flu and with COVID, right, it seems like, okay, the vaccine kind of works, but, people are still getting COVID, right? And when you take the flu shot, it kind of works, but, there's all these different mutations and variations. Is this just simply a function of the mutation speed of the pathogen, or is it that some vaccines are going to a more general form of the disease, and others are targeting kind of a more specific form of the disease?
Yeah, I think there's a range of things going on there. The first is that some pathogens don't change. So the smallpox that Jenner was immunizing against.
And did it not change because it was just so dang powerful, that it didn't need to change, or it didn't need to have any, because it discovered something that worked really, really well, so it didn't need to have like a high rate of mutation speed?
Yeah, in the case of smallpox, there's different types of the way that viruses transmit their genetic material. Some have DNA and some have RNA, which is different types of genetic material. But the ones that we call DNA viruses, the way they tend to escape the immune system is by making proteins that shut down the immune response. So they have much bigger, much more genes, and the majority of the genes are dedicated to turning off the human immune response. And so they escape by like turning off the immune response, whereas something like influenza virus or SARS-CoV-2 escapes the immune system by changing its coat all the time. So it says, now you see me, now you don't. So a vaccine that works against a DNA virus is basically looking at an unchanging thing. So the vaccine that you used in 1850 is the same vaccine you can use for the 2000, because the thing you're targeting isn't changing. with the viruses that are rapidly evolving, they are changing so the immune system recognizes a very specific bit of a protein, and if that protein changes, then the virus is no longer seen by the immune system in the same way, so your protection wanes. The other aspect to it is, and that's about how viruses escape and vaccines and the immune response, but the other part of it is why, the question was, well, why are we still getting infected even though we've been vaccinated? And there are probably 4 layers to what a vaccine can do. The perfect vaccine, or in some, the very best top level, is what we might call sterilizing immunity. And that's imagining you're basically wrapping a body in cling film so no virus ever gets in. You never get infected at all. That's incredibly difficult to achieve. The next level is that you get infected, but you clear the infection super quickly, asymptomatically, and importantly, you don't transmit viruses to other people. Below that, and a really important role for vaccines, is that you don't get disease, and below that is you don't die. Vaccines that prevent disease and death are incredibly important public health tools because they're the things that clog up hospitals. People don't want to die. So they may not be, people might be getting infected, they might be getting mildly sick, but they're not getting severely sick and they're not dying. So that I think some of the narrative around vaccines was maybe it was presented wrong during the pandemic and saying, this is going to stop you ever getting infected. And what we should have been saying is, this will stop you getting disease. This will stop you dying. This will stop people dying of this. And if people stop dying, things can go back to normal.
So, with COVID, I mean, will we get to a place where we will no longer need to vaccinate people, right? Either because of the evolution of the disease itself or because everyone will have been exposed to it at some level, right? I mean, there are a lot of people that would say if you got exposed to a coronavirus in your youth, then you have the tools in place to defend yourself against it if you got it later in life. So now that it's kind of endemic, will we be able to kind of discontinue our vaccination campaign at some point? And we've got these other 4 coronaviruses that are endemic and we don't have vaccines for them, and they don't seem to be terribly fatal for most people.
Yeah, I think it will be a decision based on how much disease is it causing in the most at-risk groups. So if it's still, I think it's probably going to stay. So I don't think it's going to disappear. But if it's still changing and still causing disease in at-risk people, they will still vaccinate. And I think it's just a balance of how much disease is it causing. It could potentially be like the other human coronaviruses and become less pathogenic. There's no kind of evolutionary narrative that says viruses have to become less pathogenic. They just are what they are.
Do we know whether the other coronaviruses are in fact relatively harmless if they were to infect someone who was immunologically naive and in a vulnerable position? So if someone were living in the plastic bubble, until they were 80 and they got exposed to the common cold virus, would that be as harmful, say, as COVID was?
Yes, I think so. I think some of it was a way of looking at what happens when a new virus emerges. It kind of gave us an insight of what would happen when a new virus emerges, and the people most at risk. It doesn't have to be particularly pathogenic. There are people who have, frail, damaged lungs, and you put a virus into there, it's going to cause disease and death.
Mm-hmm. And so, in terms of the new types of vaccines that might be on the horizon, what other sorts of things can we do? Particularly this new technology that sits behind the COVID vaccine, right? What does that open up in terms of how quickly we can start manufacturing? Would this be targeting specifically new diseases, right? Or are there old diseases that this new technology will be able to tackle besides, say, RSV. Is RSV now being are the same tools that developed COVID being used for RSV?
No,
Or?
so RSV is being made in relatively more old-fashioned technology using a protein. So it's a protein vaccine. Um, I mean, the huge strength of the RNA vaccines is the speed of manufacture from a novel pathogen. The fact that it took 60 days or 50 days from the sequence being emerging to a clinical trial beginning is extraordinarily fast. So I think their huge strength is in pandemic prevention. I think maybe once they've got into somebody, the immune response to an RNA vaccine isn't wildly different to a protein vaccine. So once you put it into somebody, you're kind of making the same type of immune response. So I don't know whether, like, qualitatively they're going to change anything. What they can do, where they could be really useful tools, one is experimental medicine. So you could say, we don't know what part of the HIV virus to target. We don't know if we make subtle mutations, can we make vaccines that cover more HIV strains? And the best way to interrogate that is by looking in people who, the best model of a cat is a cat, the best model of a human immune response is a human immune response. So you could do quicker studies where you're making multiple RNAs, which would be cheaper to do, and to test the evolution of something. The other area where I think they may be very beneficial is in individual cancer vaccines. And there's a great paper from pre-pandemic where they sequenced somebody's cancer, somebody's tumor, and they said, these are the neoantigens that are unique to you. We'll make a vaccine that recognizes these neoantigens out of RNA, and you can then treat the cancer by going from the sequencing, which is being fairly routinely done on tumors now to identify what they are, what the mutations are, to choose what drugs you can use, and then move that into vaccination. So I think it's that kind of speed and plasticity of the platform is where the RNA has real strength.
Now, how would that work? Because presumably, if you already have the cancer, then your immune system already observes it. So how could the application of a vaccine post, quote, infection make a difference?
So cancers wrap themselves up in kind of immune dampening things. So most of the time, so you might have potentially tumorous cell. One might have potentially tumorous cells in you all the time, and your T cells recognize them as being wrong or not self, or not being quite right, and kill them. But a malignant tumor will have kind of embedded itself in an environment to turn off the T cells. So what a vaccine might do is pep up your natural T cells and get them to fight the cancer. And we've seen the principle definitely works, because the new incredibly effective anti-cancer drugs, the PD-L1, the antibodies that are being used, what they do is they take the brakes off the immune system, and then the T cells start attacking the cancer. So definitely immune therapy for cancers it works. And if you can kind of train it through a vaccine, maybe that's a different way to the kind of immunotherapy drugs.
Now, we've all been talking about how, turning the immune system on can help, but aren't there situations where you want to kind of turn it off, right? I have a colleague who received an organ transplant, and they turned off his immune system to make it work, which opened up the door to all sorts of other bad things. Are there ways that we can start thinking about kind of ramping up or ramping down the immune system in a very precise way, so that we can start doing more things like organ transplants?
Definitely people are thinking about it. I think it is,
because it seems like you have broad spectrum antibiotics which just wipe out everything, and then you have these broad spectrum immune suppressors which kind of suppress everything. Will we be able to kind of do this in a more fine-tuned way?
Yes, and I think even the class of drugs that your colleague will be on now is different to what it would have been 40 years ago when they first, the breadth of the response is going to be better. One area, for example, where people are trying to retrain the immune system is in allergy. And if you can, people who are allergic to cat or peanut allergies, you can drip a tiny amount of peanut underneath somebody's tongue, and it begins to retrain the immune system to not see it as like an anaphylactic shock, but rather just as to tolerate. So there may be ways to kind of tolerate and retrain. There are also lots of treatments now to where we know the effect of like what's going wrong. We know that a cell is producing a certain molecule to attack the rest of the body. We can get rid of that molecule through antibodies, and that reduces the immune response. So that's kind of retrain or turning off the immune response. It maybe not retraining. I think the next step is like, can we reset? Can we identify the T cells that are causing problems and remove them?
Mm-hmm. Now, towards the end of the book, you talk about some of the, I guess, sociological or psychological factors that have maybe stood in the way of diffusing some of these ideas. You point to kind of anti-vax movements and so forth. And it seems like they only thrive in an environment where people are relatively healthy and safe, right? So how could we educate people besides getting them all infected? Right? I mean, if we kind of give people some malaria, give people some measles once in a while, it's like, okay, fine, I get it. Now I see the idea. Like, how can we get people to appreciate the importance of prevention when they haven't really seen some of the bad consequences of not using prevention?
Yeah, it's a perennial puzzle. And I think it's through conversations like this. It's just through reaching out. It's engaged conversation and not just shouting at each other. It's about dialogue rather than kind of aggressive kind of shouting at each other. I think it's the only way. It's about kind of, and a lot of it's coming to worldview and like how people are approaching their life. It's got mixed up in an awful lot of other things.
I mean, is there? I mean, there seems to be some trade-offs associated with acknowledging trade-offs. There seems to be some trade-offs associated with acknowledging lack of perfect knowledge, right? So, on the one hand, if people say, there's no cost to this, right? When we know that there's an extraordinarily small number of people who will have, say, adverse reactions to a vaccine, right? So to not acknowledge that can help lead to a loss of credibility. But to acknowledge that is to provide sort of the germ of an argument that would, take that out of proportion. So do you think scientists as communicators, I mean, do they need to be, more transparent in some ways, maybe more humble in some ways? What are some of the ways that scientists can become better communicators?
Yeah, and certainly like less dogmatic or more. We've been talking at work about like the role of doubt and the importance of doubt, and I think some of it got a bit muddled and everyone saying follow the science, but science or people attacking scientists or the whole process is about doubt and questioning, and that's very hard. So that strand of like what science is, of like asking questions, not being certain, is very difficult in this kind of other arm where you're like, if you do this is probably going to be good for you. It's quite a hard pitch to say, I think you should do this, but there is risk. Whereas you guess the strident other person, if you're not constrained by science, you can just say, don't do it. That. Yeah, I don't know. It's a real challenge in science communication. We have to be open and honest about what science is and means. And I saw something. It was kind of attacking the process, it's like, but we don't know these things, and it was kind of, they're like, well, yeah. I couldn't articulate the difference between scientists and and I think what it was somebody saying, science is truth. That was their kind of position. It's like, that's the science. This is truth. And it's like, well, no, it's not. Science is questioning and taking our knowledge and expanding as we go along. And I think that's true for, all academic disciplines. History is the same. It's just kind of facing the opposite direction. You're trying to piece a puzzle together in either way as best you can. And I think I think there does need to be humbleness. There needs to be saying, we don't know. But the risk then comes. It's like, well, you don't know, so I won't do it. and yeah, it's very, very difficult.
I mean, could we maybe be better at explicitly acknowledging what burden of proof we need in a specific situation? And I guess, to that point, in times of crisis, should we be prepared to accept a lower burden of proof? Do you think going forward, I mean, for the next pandemic, will our rules say of, clinical trials maybe be adjusted or need to be adjusted?
So that's interesting. I actually, the clinical trials and the burden of proof for the vaccines was the same as it was outside the pandemic. There was nothing different. It was done quicker, but it wasn't done quicker. The trials, the gaps between the trials was shorter. So normally you do the first part, you pause, you publish, you get more money, you do the second part, and it was the pause and publish was disappeared. So all the trials were done as stringently as they would normally do. The big challenge was that suddenly you had this drug that was being given to 1 billion people, and if you gave apples to 1 billion people, some people would get sick. If you gave aspirin to 1 billion people, a lot of people would get sick. If you gave paracetamol, anything to that many people that quickly, some people are going to get sick. And I think that communication of it's hard, right? Say for most of you this is fine, but for some of you, and we don't know who, some of you are going to get very sick, and that's a tricky sell.
Well, I mean, if the severity of COVID was like that of the 1918 flu, I mean, presumably we would have a lower burden, right, of proof because of it.
No, I disagree. I think we'd have stayed the same. We would have gone into the like the NPI, the non-pharmaceutical, the lockdowns, the hand wash. That would have been much stricter, much sooner. I think the medicine and the drugs don't change the way that the safety, the way something like the FDA approaches these things doesn't change, but the what's happening societally wise would change to manage the risk quicker.
Mm-hmm. Well, John, thank you so much for joining me. The book is Infectious. I mean, I don't think we did justice to the entertaining aspects of this book, which I know you had a lot of fun writing. And of course, it ends with you getting infected, which of course, it has to. Right? And so you talk about how, you waited for it and you managed to get most of the book done before you managed to get COVID. So appreciate you joining me. This book, check it out, Infectious. It's not, by the way, it's not a book on COVID. It is a book on infectious diseases, on microbiology, on immunology, epidemiology, all the fun stuff that you wish you knew like 3 years ago. Hope more people will read it. Let's talk again soon.
Great. Thank you very much.
unSILOed podcast is produced by University FM, elevating the stories of your institution.